Hey, Quinn. How are you guys doing?
Good.
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 So, we technically have Friday off, so I will not be seeing you on Friday. According to the provost, we're supposed to take that day off. So don't do anything school related, as if you were on spring break. I don't know how you feel about that, but I really think I'm going to miss my spring break this year. How are you guys feel about the whole situation?
I mean, at least they still gave us some days off. Like my sister's school, they just canceled spring break and didn't give them anything. But the assumption that I'm not going to be able to do anything on Friday is probably a little bold.
Mm-hmm.
Okay.
I'm going to have
well,
stuff I have to do.
You're gonna have what?
I'm going to have stuff I have to do.
Oh yeah, like what?
Reading. Well, we have conference next week, so I'm trying to get ahead. So that's
Okay.
kind of for this time.
Yeah. So there are times when you take spring break and you use part of that spring break to kind of catch up on things and get ahead on things, but this seems like a really disjointed sort of way of approaching spring break, because there's something about just kind of having a break in the middle that allows your brain to reset a little bit, as opposed to, rather than going, so we have a 16-week semester, going 8 weeks and taking a week off and then going 8 weeks again. Now we're just going to be doing 16 weeks altogether with just little tiny respites spaced here and there. So one of the things that you should think about as you kind of approach this semester differently than you've approached other semesters is know that you're not going to get that long mental reprieve that normally comes in spring break. I didn't realize how important that was for me during the fall when we have fall break. Much shorter little break, but it's like a Thursday and a Friday and then the weekend. And it's like, oh, from Wednesday when I get out of class, I don't actually have to be back in class until Monday. And when I first got the schedule for the fall, I'm like, oh, well, that's not going to be a big deal. And then I got to the end of the fall, I'm like, okay, fall break's more important than I thought it was. So just kind of from a mental health standpoint, and also from the standpoint of how you learn most effectively, seldom do people learn effectively by just constantly pushing, push. Studies have shown, I found this true in my own learning when I was a student, breaking information up into shorter bite-sized segments more frequently is better than like saying, I'm going to sit here and I'm going to study this thing for 3 hours straight. So pay attention to how you go about learning typically, and put that up against kind of the way the schedule is operating this semester to say, okay, how am I going to get through these 16 weeks where there really isn't any extended break or respite during it? Start planning for that now so that you don't get to week 12 and you're like, oh my God, I'm dying. Because it is going to be a different feel, and it's going to feel worse as you get further into it probably. So begin to kind of structure how you're studying for all of your classes of this one, how you're balancing your workload in ways that will get you through it successfully. And that's going to be different for everybody probably. Those of you who are seniors and are also working on senior research, that's another consideration that you need to think about. Think about, okay, when are things due during that progress of things? Rose, you have your preliminary data presentation coming up next week, don't you?
Yeah, it's on Monday, and Dr. Reynolds and I are meeting on Friday to practice.
Oh, on Friday, so you guys are not taking the day off. Okay. So
Yay.
yeah.
Can't wait.
yeah, so just basically take care of yourselves and think of this not as a sprint that you can power through. Think of this as a marathon where if you just slow down a little bit, you'll make it through. The goal of any marathon, have any of you ever run a marathon before? I ran a marathon when I was in high school. My goal was to finish. 2 summers ago, I rode in this thing called the Triple Bypass in Colorado in 2019, summer of 2019. Have you ever heard of this?
Yeah.
Triple Bypass. It starts in Evergreen, Colorado, and ends up in Avon, Colorado. It goes up Squaw Mountain, then it goes up Squaw Pass, and then it goes up Loveland Pass, and then it goes over Vail Pass. It's 117.5 miles, and it's 10,000 feet of climbing. And my goal, it was my first time riding in this event, and my goal was just to finish. And I rode with 3 other guys, and 2 of them, their goal was to do better than they did the last time they rode it. And 2 of us, it was our first time going, and our goal was to finish. The 2 guys who had the goal of finishing finished. The 2 guys who had a goal of beating their previous time, they dropped out at mile 52. Think of this semester more as a marathon or a triple bypass. So anyway, as we approach our first spring break day, be thinking about that, be aware of that, because it might be the difference between finishing the semester successfully and finishing the semester less successfully than you might.
I just want to finish.
You just want to finish. Yeah. I mean,
I don't really care how at this
quite
point.
frankly,
Just want to graduate.
that's a reasonable goal. You know, just finishing successfully, as opposed to the only alternative to not finishing successfully is not finishing.
I straight up do not want to come back here next fall.
Okay, well, good. Unfortunately, I'll be back here next fall. So, you know, if things go well. All right, questions about other things. I realize I gave you an assignment, and it's not going to be a graded assignment, but I still expect you to do well on it. But you're seniors and juniors, and so, well, all of you are seniors. The only junior is not in the class online, so you should know how to perform well when you're not being graded. The main reason is I feel like I'm doing a lot of the talking, and you guys are just sitting there, and I want to get you guys more engaged in it. And I put this in the email. So expect kind of more of that sort of thing, things that I'm going to do to ask more of you to try and keep you engaged with the material. So what I'm going to do today is I'm going to introduce the frogs and toads, and then we'll start working our way through the families, and we'll just see how far we get today. Once again, the goal of this stuff here at the beginning is to orient you towards the organisms that we're studying in the class, so that when we start talking about their physiology and their behavior and their reproduction, when I say, oh, the caecilians do this and the anurans do that, et cetera, you have an idea of what those organisms are. So that's the purpose of these days, when we're looking at a broad overview of what the frogs and toads are like, what are the salamanders like, et cetera. So I gave you the frog families that I think are going to be important for you to know something about. And then I'll cover the caecilians, because the caecilians are super unusual. We don't even have them in the United States, but it's a group that you should be aware that exists. So that's why we're doing it, and why we're doing it now rather than at some other time during the semester. So can you guys see my PowerPoint slides?
Yeah, I have a quick question first.
Yeah.
Can you upload the daily schedule to Moodle, or at least just the exam dates, just so I can plan my life out?
Yes, I've been meaning to get that done, and I just had other things on my plate. But yeah, it's mostly finished. I'll try and finish it up soon.
Okay, thank you.
All right. Part of the issue has been working in research around the schedule. So I'm trying to, it took me a little while to figure out when I can go and do some field research with Zach. And so that's part of what's been delaying me getting something out.
No, that's
But
fine.
now I think I figured that out. Not having a spring break, the other thing that's done is it's basically made it more difficult to go and do field work in Texas, which is kind of where his field sites are, Texas and Oklahoma. So we're going to basically cover 3 field sites in 2 different states in 4 days in the end, working around the weird spring break days off thing. So, all right. So frogs. Why is sharing paused? You would think I would be better at this. Okay. See a big frog skeleton? Yeah, okay. So frogs and toads have a lot of skeletal modifications that are essentially related to the fact that they locomote using saltation. They jump from place to place. And so one of the things that you'll notice about frogs is they will oftentimes have stout bones in the forelimbs, long bones in the hind limbs. They have a reduced number of vertebrae. The presacral vertebrae are reduced in number, 9 or fewer. And the sacral vertebrae are all modified and fused into this thing called the urostyle. I think it was Elizabeth Ellis who was asking me about the Plains leopard frog, and she saw the ilium and the urostyle of the pectoral girdle that was kind of poking out on the specimen because it had kind of dried out and shrunk a little bit when it got preserved, because one of the things that ethanol does is it sucks the water out of the specimens, which is part of the way that they stay preserved. And she was mistaking the ilium for the dorsolateral ridges, but really she was just seeing the ilium and the urostyle through the skin. And so the urostyle is basically the sacral vertebrae that are all fused into this modified bar of bone here. You also see that there is fusion of the tibia and the fibula. And so the reduced number of vertebrae, the fusing of sorry, did I say sacral vertebra? The fusing of the caudal vertebra into the urostyle, and the structure of the bone, the fusing of the radius and the ulna. These are all modifications that essentially stabilize the skeleton as these animals jump, and when they land, there's a lot of force applied to these bones in the process of landing. And so you have the bones fused and less flexible, and certainly the vertebral column is less flexible. You have reduced cranial elements that are also probably part of that, but they also might be related to feeding adaptations. So a lot of skeletal fusion. The bufonids, the true toads, don't have any teeth, although the rest of the groups have pedicellate teeth. And the vertebral structure is very variable, and this is shown on the next slide, but we have 3 different types of vertebra within the frogs and toads: amphicoelous, opisthcoelous, and procoelous, and those are illustrated on the next slide. So this is the amphicoelous condition. When you think about the vertebra, you can think about the vertebra enclosing both the nerve cord and a notochord. So when you were an embryo, so you're a member of the phylum Chordata. These are the chordates, and they're called the Chordata because at some point in your life you had a notochord. Now, in us, that's something that occurs in us when we're embryos, and over time, the notochord is replaced by the centrum of the vertebral column. But in fishes, for example, the notochord stays intact throughout the entire lifespan of the animal. So, for example, if you took a comparative anatomy class and you dissected a shark, you would be able to cut through the vertebral column, and inside the vertebral column, you would find a notochord encased in the vertebral column. And in the case of frogs and toads, we have some frogs and toads that have the notochord still intact, but in many of the frogs, we have the notochord that has been replaced by the centrum of the vertebral column. So the notochord has been completely replaced by bone. And so in the amphicoelous condition, which is found in the most primitive frogs, we have a vertebral column that is not fully ossified, and it still has a notochord. And as a result, there really isn't a joint, joint of articulation between one vertebra and another vertebra. And then when we have the notochord replaced by completely ossified centrum, we have two forms of articulation. One form of articulation is basically when we have the procoelous, coelom, yeah. Procoelous, where there's a cavity at the front end of the vertebral column, and there's a bony projection at the hind end. And then opisthoscoelous, which is where there's a cavity at the back end of the vertebral column, and there is a bony element that fits into that cavity at the front of the vertebral column. And this is confusing terminology. So procoelous and opisthoscoelous if you are up on your Latin, pro means before, or sometimes front, sometimes translated front, and coelom is basically hole or opening, or in this case, you might think of depression, not the emotion of being depressed, but a depression like a depression in the ground or something like that. And so in this case, procoelous basically means that the opening or the depression is at the front of the vertebral column. And so here you see in this articulation, there's a cavity here, and then the centrum inserts into that cavity and forms the articulation. Opisto basically means hind. The hind part, the posterior part of the body, and once again, the depression is on the hind part of the the vertebra, and you can see that in the opisthoscoelous condition, you have the cavity on the back side of the vertebra, and the articulation of the bony part that articulates inserts into that rear-facing depression. So procoelous, don't think, oh, procoelous means that the centrum sticks out in the front. It isn't the centrum sticking out, it's where the depression is, procoelous versus opisthoscoelous. So the terminology seems a little backwards there sometimes. This becomes important for taxonomy. So some families will have the amphicoelous condition, some families will have the procoelous condition, some families will have the opisthoscoelous condition. Both the procoelous and opisthoscoelous are found in more recently evolved groups. Amphicoelous is believed to be an ancestral condition and is found in the earliest frogs and toads. So frogs and toads in general have characteristics of their mating and reproduction that vary taxonomically as well. And so when they go to reproduce, they engage in what is called amplexus. And so these are different frog groups involved in amplexus. And in this case, this is a leaf frog from, I think, South America. And you can see that the large frog here is a female and the male is much smaller. So there's sexual dimorphism in this case. And in this case, these two individuals are in amplexus. And the reason that you enter into amplexus with a female as a male is that as she ejects her eggs out of her cloaca back here at the back, as her eggs exit, the male is going to basically expel his sperm over the eggs, and that's when fertilization occurs. So in this case, fertilization is external except in the tailed frog. And in this case, the male has grasped the female around the base of the hind legs. This is called inguinal amplexus, or inguinal amplexus, depending on how you pronounce it. And that basically refers to grasping them around the body where the legs attach to the body. We have axillary amplexus, which is basically when the male grasps the female underneath the arms. That's where axillary comes in. And then cephalic amplexus is when the male grasps the female around the head in front of the front legs rather than behind the front legs. And in some cases, cephalic, I think, is the least common amplexus type. And in some cases, frogs do this where the male frog is actually substantially smaller than the female frog. He'll be on her back, and then the female frog has a groove in the body that goes down the back. And as he expels his sperm, it basically gets channeled down this groove so that her body channels the sperm right to where the eggs are going to be exiting the female. And so this becomes important both behaviorally and also these different forms of amplexus are often found aligning with particular families. Particular families will have a certain type of amplexus. Once they lay their eggs once again are usually but not always laid in water. They produce a larval stage. In most but not all cases, those larvae are free-living larvae. In direct developing species, the larvae remain in the egg through that entire larval period. They absorb the tail, they grow the legs, and they emerge from the eggs as little miniatures of the adult. So this is a very old figure that I just kind of like for historical purposes, showing the life cycle of a frog. And so when the frogs hatch out and they're free living, they generally begin as tadpoles. In most cases, they don't have external gills, they have internal gills. And as they grow, they will generally sprout hind legs first, and then over time, you will start to see the forelimbs bud out as forelimb buds. And over time, they slowly develop the shortened forelimbs and the longer hind limbs. And as they're doing this, they're also slowly resorbing the tail. So the tail will generally get smaller and smaller. When they hatch out, they will generally be a little hatchling with a little nub of a tail. And then they will continue to resorb that tail after they've emerged from whatever body of water they were a larva in. And then they will take on the adult form. Tadpoles vary a lot in their morphology, and you could spend a lot of time just thinking about larvae. And two guys that I admire a lot, Ron Altig and Roy McDermott, they've produced an entire book about the larvae of frogs and salamanders in the United States. And maybe I'll bring this up to lab so you can take a look at it. But they have gone to a lot of trouble to try and take photos of and make detailed drawings of all of the larvae of all of the frog and salamander species that occur in North America. And so there's plate after plate after plate of larvae in this book. And one of the things about larvae that is important is the denticle structure. So I should have put this in the PowerPoint, but right around the mouth here, you can see that there are these cornified structures that they use for feeding, and they're located right here. And so whether the mouth opens to the bottom or whether the mouth opens more in front, the structure of the denticles, the structure of the fin, et cetera, these are all diagnostic in identifying larvae. And if you have a larva and you come from a part of the world where there's been an effort to describe what the larvae look like, you can actually identify species of frogs just by looking at the larvae. In this case, you can see that there's a siphon right here on the side of the head. This is where the water exits the animal's body as it's running over the internal gills. In some cases, the siphon is located at the front of the head. In some cases, the siphon is located further back. You can oftentimes see the intestines through the body, and so they're semi-transparent. And all of these things are important in identifying larvae to species. Oh, I do have denticles in here. So denticle shape and placement are important taxonomically, and whether you have a siphon that exits on the side versus one that exits medially, and whether it exits medially at the rear end of the tadpole or at the front end of the tadpole. And then in most cases, if the siphon is lateral, there's a siphon on both sides, but in some species, there is a siphon that only exits on one side. And so once again, these are called type 1 through type 4 siphon placements. You'll often find that siphon placement is correlated with different kinds of mouth structures as well. Most of these larvae are grazers. They tend to feed on algae that are growing on the surfaces in the areas that they live. Sometimes they will actually dredge up mud and feed on algae and things like that are in the mud. In some cases, like in the spadefoot toads, they can have highly modified mouth parts that start to resemble actual teeth when they transform from a more algae-feeding morphology to a cannibalistic morphology. In these explosive breeders that are in ponds that dry up quickly, and there's this kind of race to transform into the toadlet before the pond dries up. The easiest way to gain mass, because metamorphosis from the larva to the toadlet is determined by the mass that you achieve. If you're stuck in a pond that is drying up and the pond is full of a bunch of other spadefoot toads, the easiest way to gain mass is to eat other spadefoot toads. But the normal morphology of a tadpole doesn't really allow you to do that. And so in those conditions, they have a phenotypically plastic transformation that occurs that transforms them into this cannibalistic morphology that then allows spadefoot toads to eat one another. And pretty much the more spadefoot toads that you eat, the faster you grow, the earlier you metamorphose, and you escape the pond before the pond dries up. If you don't escape the pond before the pond dries up, and the pond dries up while you're still a tadpole, then it's game over at that point. So this is the phylogeny that is in your book for frogs and toad species. Once again, time in this case is going from the bottom of the graph to the top of the graph, and then evolutionary diversification is on the x-axis in this case. And I'm not going to ask you to reproduce this or anything else, but one of the things that you might want to do with this figure is notice that the relationships among the different families in many cases are not very well resolved. We know that all of these families are grouped together, but we are not clear about what the relationship is, for example, between the Bufonidae and the Hylidae. We don't have any idea whether the Bufonidae are more closely related to the Hylidae, or if the Hylidae are more closely related to the Hemiphractidae. So whenever you see this thing, which is a polytomy in a phylogeny, basically, we know that these are all grouped together, but we don't know what the relationships are within that group. We know that these are all grouped together, but within that, we don't have a clear idea of what the evolutionary relationships are. So frog systematics is an active area of research. One of the reasons that, for example, the tree frogs that you're looking at in lab have changed genus. So the tree frogs in North America used to be in the genus Hyla, and they've actually been removed from Hyla and they're placed in a different genus, Dryophytes, is because of, once again, a taxonomic revision of the Hylidae in North America that was published since the last time I taught this class, which was 2 years ago. When I was an undergraduate taking herpetology for the first time, all of the species that are in the species Lithobates now were, all of the species that were in the genus, that are in the genus Lithobates now, were in the genus Rana when I took this as an undergraduate. So taxonomy constantly changes because taxonomy is constantly being examined from the standpoint of trying to resolve these relationships. So, Kiara, I believe, was it Kiara? You had a Scaphany?
Yeah, it's me.
Yeah. Do you have something prepared, or are you just going to talk and use the slides that I already have done?
You can just use the slides. I
Okay.
just have some stuff on a Word document, and then if
Okay.
somebody wants to follow along in their book too, it's on page 61.
Yeah, and I'll just, like I said, I'll just fill in any gaps that I think need to be covered.
Okay. So the first thing, I kind of just went along with the book and what they had for certain characteristics and stuff like that. So they're unique frogs, obviously. They have an intermittent organ that resembles a tail, which is why they're called tailed frogs. And this tail is highly vascularized, and it's extension of the, and if I'm saying this wrong, it's a cloaca.
The cloaca.
Cloaca.
Yep.
And that's supported by cartilaginous rods that is attached to the ventral part of the pelvic girdle. Ascaphus has 9 vertebrae, and that differs from 8 or fewer from other frog species. Let's see, what else do we have as important?
So what does that mean in terms of how old this taxon is in terms of evolutionary age? Is this one of the earliest frog groups or one of the later frog groups?
Later. Right.
Earlier.
Earlier. Okay.
Yeah. Evolutionarily, what's happened over evolutionary time is they've lost vertebrae. So you tend to find more vertebrae in the more primitive frogs and fewer vertebrae in the more advanced frogs.
Okay. Well, then that would make sense for my next thing is that they have a prepubic element and the epipubis, which is not found in other frogs, except like Leoplema, Xenopus, and I don't know how to pronounce the last one. It's like Sudi Hymenicrus.
Yeah, so you have the Ascaphidae here. You have the Hypipede, which is where Xenopus is. These are all the earliest evolving frogs. They're at the base of this evolutionary tree. Yep. Yeah, so all those characters that you just mentioned, those are thought of as being primitive characters or ancestral characters. Continue.
And then for the habitat, they inhabit cold terrestrial streams and are highly aquatic. And on page 61, it kind of just shows their geographical range. They distribute in northwestern US and Canada and through the Rocky Mountains. It's a pretty small range from other frogs that we've seen so far. Their eardrums are absent, and I found this kind of interesting. The frogs don't call, which is kind of weird. You know, when I think of frogs anyways, I think of frogs calling, and so I just thought that was an interesting characteristic.
Once again, so is frog calling a derived character or is it an ancestral character?
Well, if these frogs were earlier, they would not be calling, so it's technically old, so ancestral.
So not calling is ancestral, and then calling would be
Derived.
derived. Yep. Mm-hmm.
And then for reproduction, they have inguinal amplexes that occur underwater, and they lay a few large eggs under rocks in water, and then the embryos develop very slowly.
Did it give any reason why they developed slowly?
No, it does not say.
Okay, so these guys are montane in terms of their distribution. They occur up in the mountains. They're subjected to cold temperatures generally, and so the slow development is probably related to the temperature at which they're developing because of the temperature of the streams that they're found in. What else do you have?
That's basically all I had. I mean, the 3 points that you have are the most important, I think. I would say the vascularized extension, internal fertilization.
Yeah, but you did the same thing that I do, which is I put some things on the slide and then I add 2 things as you're going through their aspects of reproduction and stuff like that. When you guys, this is not for Kiara, but anybody else, when you look at this geographic range, what does this geographic range remind you of? Have you seen this type of distribution before?
It looks kind of like a ring species
It looks,
without the upper arch, though.
without the upper arch, but have you seen the same pattern in any other groups? So there's Rhyocotriton, which doesn't have the Rocky Mountains aspect of it. And then we have the Dicamptodonidae, which are the Pacific Northwest, and once again, the Rocky Mountain representatives. This is very similar to the distribution of the Ascaphidae. And then in terms of the salamandrids, at least the salamandrids in the Western United States, once again, you have this coastal population, and then you have these relic populations in the mountains of the Bitterroots and the Rockies. And so in all of these cases, these mountain dwelling species were probably more broadly distributed in the geologic past. And as, once again, the climate has dried out, the connections that probably used to connect these 2 things, dry habitats have intervened in the middle, such that they're now only limited to these montane areas that are cooler and moister and have the types of habitats that they're going to be breeding in, these, once again, these clear, cold mountain streams. So this is a repetitive sort of distribution that you see both in salamanders and frogs in the Western United States. If you've ever driven from like Missouri to Oregon, one of the things that you'll find is you go through the Rockies and you're up in nice cold mountain areas, and then you descend into the northern reaches of the Great Basin Desert, and there's this really pronounced dry area until once again you get to these mountain ranges over near the coast that are considerably rainier. And so there's a rain shadow effect, and that rain shadow effect basically creates a geographic barrier that separates these two parts of the distribution of the family. All right, good, nicely done. Who was next? Who had the Elytidae?
I do. So it's a family with 3 genera and 11 to 12 species, depending on which source you're looking at. They're called the midwife toads and disc-tongued frogs, and they're found in Western Europe and North Africa. And the midwife toads represent an exception to the rule that the frogs lay their eggs in the water. So instead, mating and fertilization will happen on land, and the male will then carry the eggs on his like lower back and hind legs, as shown in the photo. And he'll keep them until they're ready to hatch. And at that point, he'll deposit them in the water.
So why did I include the midwife toads in this treatment of looking at the diversity of frogs?
Because they represent like an exception to the complex life history rules that like the frogs lay their eggs in the water.
Yeah, you're never going to see these guys, but it just represents one of the interesting ways in which there's variation in reproduction. Ironic that they're called the midwife toads, but it's of course the male that carries the eggs. Should be the mid-husband toads, I guess. Yeah. Okay. Who had the Pipidae?
did. So they are also called the tongueless frog or the African clawed frogs. And so this family of primitive frogs are tongueless, and there are 41 species in the family. And from your photo, you can see that they're found in tropical South America and sub-Saharan Africa. And the males in this family lack vocal cords, but they communicate underwater via clicking sounds, which they make by a modified laryngeal apparatus. And you put the question, why would we want to know this frog? And so again, it shows the diversity, because I normally think of frogs as ribbiting and calling out, and they don't have that. But also, I didn't know if this was a reason that you wanted us to know it, because 2 members of this genus have become model lab organisms. And so I thought that would make it important to know, because they're easily maintained in a laboratory, and it's easy to induce their ovulation and mating year round for research.
And what's the size of their eggs like?
I don't know.
They're big, so they're easy to work with. So yeah, that was actually one of the primary reasons that I included this in the list of families to cover, is because Xenopus is a model organism, and it has a variety of characteristics that make it good for that. You mentioned a couple of them. Easy to stimulate them to reproduce. They're relatively easy to maintain in labs. The other reason is that they have very large eggs. So do you guys know what patch clamping is? Do you guys remember what patch clamping is from evolution and ecology? Nobody? Okay, so back when we covered the snake newt case study in evolution and ecology, the way in which they tested to see if tetrodotoxin affected the sodium channels is, remember that they genetically modified the sodium channel gene to include a big chunk of human DNA and a smaller chunk of snake DNA in the region of the sodium channel that they thought was being affected by tetrodotoxin. They then spliced that into Xenopus eggs, so that the Xenopus eggs, as they developed, would basically express that altered gene to express the snake sodium channel in a frog egg. And the reason that they wanted to do this in Xenopus eggs is because Xenopus eggs are big enough that you can do what they call patch clamping, where you take a really small glass syringe and you basically apply suction to the cell membrane of the egg, and in that part that you've suctioned down, that you've suctioned onto with the syringe, there are sodium channels trapped underneath that syringe. And then in that syringe is a little tiny electrode that measures electrical current, so that you can see when you stimulate the membrane of that egg, it will depolarize the same way a nerve cell depolarizes, and you can measure the change in electrical current in the membrane outside what's being patch clamped and what's inside being patch clamped. And the trick with patch clamping is that you have to do it on cells that are big enough for you to actually manipulate. And so one of the other reasons that people use Xenopus is that they just have eggs that are big enough that you can manipulate. So as a result of it being a model organism, they've been sequenced. We know a lot about their developmental biology. We know a lot about the genes that are activated during various parts of their development. And so it's a pretty decent vertebrate model species, although it's very different than, for example, a rat or a mouse, because they're clearly not mammals. So you've been exposed to Xenopus in the past. You just don't recall it from that section of those last couple of papers that you read back in evolution and ecology. Questions about the pipids? for Rose or myself. All right, cool, nice. Scaphiopodidae.
That's me. So it's a small family. There's only about 7 species, and they have the typical shape of a burrowing frog. And then, so they're found like within the U.S., if you can see on the map, central, and then down into Mexico and the East Coast. And they have hard keratinous protrusions on their feet so that they can dig, which I thought was kind of cool. I never really thought about a frog digging. And then they differ from toads because they have vertical pupils and no parotid glands. So I know that's been like the parotid glands have been one of the things at least I've noticed when we've been in the lab that was been in the books. So that's one thing that I just thought was interesting in looking at the different frogs and toads that we've done within the lab. And they have cannibalistic larvae, which is weird and gross.
There's a picture of a cannibalistic larva eating one of its fellow pond inhabitants in this slide. So that keratinized digging apparatus, that keratinized digging structure on their feet is referred to as a spade. And these are called, that's why they're called spade-foot toads. So it's on the hind leg. And we saw one of these, we saw a video of one of these burrowing into the soil in a terrarium the other day. Why do we tend to call toads toads and frogs frogs? So you mentioned that this is a spade-foot toad, but it's not the same as a true toad in that it doesn't have parotoid glands. Why do we tend to call these guys toads?
I don't know.
Anybody else?
Isn't it something to do with leg length?
Nope, not leg length. So where do these occur when you look at their geographic range? They occur in the southeastern United States where it's relatively moist, but where else do they occur?
Up north where it's cold.
They occur up north where it's cold, and they also occur where? In the American West, where it is
Hot and dry.
hot and dry. And so we tend to put frogs and toads into the toad category when they have generally thicker, more keratinized skin. They're less likely to desiccate in dry environments and things like this. And so if you were to pick up a spadefoot toad, you would find that they feel less mucousy than, for example, a tree frog. And the same would be true of the true toads, the Bufonidae. So they just generally have thicker, less mucousy skin. And that's why they're informally called toads. But they're not, don't confuse them with the true toads, which is the family Bufonidae. They tend to be explosive breeders in many cases. So once again, when I refer to this term explosive breeding, I refer to a group of frogs or toads that breed over a really short period of time in these gigantic aggregations. And then once that is over with, breeding season is over. And so there won't be any toads on the landscape, and then it will get this big torrential rainfall, and then there will be thousands of toads on the landscape, and then you'll come back the next day and there will once again be no toads visible in the landscape. There will just be tons of eggs in these temporary pools. When I was in graduate school, my office mate got married, and he's from El Paso originally. So I flew out to El Paso to participate in his wedding. And we got there right after they had had one of these big torrential monsoon kind of rainfalls in El Paso. And in the neighborhood where he lived, there was a public park, and this public park had been built on what is a big clay-bottomed shallow pan of land. And it was probably, I don't know, 400 meters by 400 meters, this park. And the rain had filled that park such that it was just this gigantic, really shallow pond, probably about this deep. And he told me about this. And so while I was at his parents' house waiting for lunch to be made one day, I hiked over to this pond, to this park that had basically been converted into a pond by these rains. And I started wading into the pond. And of course, the reason he told me about this is he knew that I would want to see this. But as I walked into the pond, I began to see the water ripple out in front of me. And as I walked further into the pond, the water was rippling all around me. And it was basically millions and millions and millions of spadefoot toad tadpoles. And they were basically swimming away from me. And after I passed, they were swimming up against me. And so you could just kind of feel these squishy, mucusy things going by your ankles as I walked through this pond. And Rose is like, why on earth would you want to do that? But the thing of it is, when that thing is only this deep, that pond, even though it was very large in total area, was only going to be around for a couple of weeks before it totally dried up. And so this would have represented, if I had been able to stay there for 2 or 3 weeks, this would have been one of those environments where there's a race to develop and get big enough that you can go through metamorphosis. And this would have been one of those places where you would see these cannibalistic larvae developing, start consuming other members of your own species in this gigantic pond before that very shallow, very large surface area pond just finally dried up into nothing. And I never went back there after that was done because there was no reason to go back to El Paso just to see a bunch of frogs. But I wonder if after that happened, how many of the tadpoles actually didn't make it out of the pond before the pond dried up. And one of the practical, pragmatic things that I thought about is, I wonder if that's a cleanup issue for the parks department in El Paso. Like, oh, now you have all these crusty, probably rotting tadpoles that didn't make it out of the pond. But it was really impressive. One of my memories of grad school that sticks with me is just the sheer number of spadefoot toad tadpoles that were in that park that day. So that's what I mean when I say explosive breeder. Tens of thousands, hundreds of thousands, and millions of tadpoles produced in a very short window of time. Questions about the spadefoot toads? They do occur in Missouri. Speyer bombifrons occurs down in the river bottoms. We don't have any in the collection because probably nobody's ever gone out in the summer to collect them. Well, one of my goals this coming summer is to do that, actually. Records for them in Missouri are relatively uncommon. They're a little more common in Kansas. There are actually quite a few records around Lawrence. Not surprisingly, because they have a big herpetology community in Lawrence. All right, who had the Eleutherodactylidae?
I did. So they are, there's 4 genera and they have 207 species and 2 subfamilies, so it's very species rich. They were actually originally placed in I don't really know how to pronounce it, but the Leptodactylidae.
Yeah, the Leptodactylidae.
And they've recently become their own family instead of a subfamily. And they inhabit central Texas through northern South America and the Caribbean. Their sizes range, as he has on his slide. For some of the species in this family, they have internal fertilization, and there's no free living tadpole stage. The eggs develop directly into small froglets. And yeah, they do Amplexus axillary.
So this is not a family that occurs here in Missouri. There are some representatives that occur, once again, you mentioned in central Texas, central and west Texas. That's kind of an exception to the general rule in that they tend to occur in the moist tropics. The family Eleutherodactylidae is named because the first species of this group was found on the island of Eleuthera, which is in the Caribbean, a little tiny island in the Caribbean. And so that's where the Eleuthera comes from. The main reason that I wanted you to know these is that it is among the most speciose group of frogs on the planet. And they are all, for the most part, you know, small brownish leaf litter dwelling frogs. The other reason I wanted you to know about them is because they're one of the groups of frogs that do have direct development rather than free living aquatic stage as well. They can reach incredible densities. So the species of frog that I've worked on in Puerto Rico is one of these frog species where it can reach, you know, numbers exceeding 25,000 frogs per hectare. Remember that a hectare is 100 meters by 100 meters. Stuff 25,000 frogs in that space, and that's a lot of frogs to contend with. As a result, they can have potentially big effects on things like nutrient cycling and control of insect pests and things like this. Because they're direct developing, what do you think might be one of the problems with these frogs in terms of thinking about these frogs causing problems for humans? What does direct development allow a frog to do that a frog whose reproduction is tied to water can't do?
Like they aren't tied to a body of water, so they can just put their eggs anywhere and have them survive.
Yeah, they can put their eggs anywhere and have them survive. They oftentimes lay their eggs in the phytotelmata of plants. We've mentioned phytotelmata already. These little aquatic environments that dendrobatid frogs, for example, use to house their larvae. But those phytotelmata, even if they dry out, they're considerably more moist, and so a lot of times you'll find males guarding a group of eggs that have been laid by a female, and inside the leaves of these plants, like bromeliads. And if you have bromeliads being transported on ships around the world as part of the horticultural trade, if those bromeliads have a frog with a clutch of eggs, those eggs, because they're normally not laid in water, they're more resistant to drying out. And because they go through direct development and they don't have a free living aquatic stage, they hatch directly out of the egg into a free living little frog. They can be transported around the world in ways that aquatic breeding frogs cannot be transported. Former student of mine and I studied this frog. This is one of the Eleutherodactylus frogs, and we were looking at this frog, which is normally native to Puerto Rico, and we were looking at the spread of this frog throughout the Hawaiian Islands, doing a population genetics study in the Hawaiian Islands. And the whole way that it got to the Hawaiian Islands was probably through horticultural exports and imports. Plants were picked up in Puerto Rico. They were put on a ship. They were shipped to Hawaii. The entire time that they were on the ship, there was a male sitting on a group of eggs. They went through their development on the ship, and when they got to Hawaii, they eventually hatched out, and all of a sudden you have a population of frogs in a place where there actually are no native frog species. And so these guys have the potential to become an invasive species because it's just easier to transport them from one place to another as a result of their particular development style. We were actually interested in whether or not these frogs were being moved around by human beings. And what we found is that they probably are. And even while we were there, we got in a car leaving one site to go to another site to do some collecting. And we got about 2 miles down the road, and a frog jumped out of the engine compartment onto the windshield of the car. So they're very, very adept at hitching rides on things. So Questions about the Eleutherodactylids? Yeah, the ones that occur in Texas are like the Rio Grande chirping frog. They're very unusual looking frog for a place like Texas. The cliff chirping frog, the Rio Grande chirping frog, those are all in the family of Eleutherodactylidae. And once again, you won't see them unless you go and knock around certain parts of central Texas. Who had the Hylidae?
I did.
And I think if we can get through Hylidae, that'll be the end for today.
Okay, sounds good. So the Hylidae are American and Australian frogs. They are diverse in both species and morphology. So they have over 50 genera, 947 species, and 3 subfamilies. And from the graph, you can see that they inhabit North and South America, the West Indies, extreme North Africa, and temperature Eurasia to Japan. So they range in size from less than 30 to 150 millimeters. Some of these characteristics, I'll say, like some or most have this, but like there are exceptions to some of the frog species in Hylidae. So most are arboreal with developed toe discs and claw-shaped terminal phalanges, but some live in tree holes. Phosphorial hylids live in arid areas and form cocoons to protect themselves from desiccation. And there are others that live in dry environments. And those that live in dry environments have unusual modifications to their skull morphology. And another one, most hylids are strong jumpers, but then there are some that are arboreal walkers and make slow movements. And another one, most do not have extensive macroscopic glands, but many have conspicuous glands on the dorsum. And for reproductive, their modes are diverse. Amplexus is axillary, and most hylines lay eggs in water, and then they have aquatic tadpoles.
So those species that are climbing or crawling, how is their morphology different from the hopping ones?
If they're crawling, they have the claw-shaped terminal phalanges.
Okay, and what about the relative lengths of the front and hind legs? You know, in most frog species, you have fairly long hind legs and relatively short front legs. How's that different in these climbing species of hylids?
I'm assuming if they jump, they have longer hind legs, and if you crawl, you have shorter legs.
You don't have shorter hind legs. You generally have longer forelegs,
Longer.
longer, thinner forelegs. And in this photo on this frame here, you can see the Phylomedusae subfamily, and you see this bump on the back above the eye and tympanum of this species. That's one of those dorsal glands that Kiara was mentioning that you find, even though they don't have a parotoid gland specifically, you will see regions that are more glandular in some of these groups. The toe discs, if you look carefully at the tree frogs that are up in lab, to take a look at this tomorrow, you will see that there is an intercalary cartilage. So the phalange comes down, and then there's an intercalary cartilage, and then there's the toe disc. And this intercalary cartilage, and I can't spell like always, this intercalary cartilage actually is useful in making sure that the toe pad is able to make flush contact with whatever surface it is on. So this kind of rotates around on that intercalary cartilage, because it's this toe disc which is actually going to allow it to adhere to surfaces as it's in a tree or on a pane of glass or something like that. And so that the toe disc combined with the intercalary cartilage is one of these adaptations for this arboreal lifestyle that they have. Questions? All right, we'll finish off the frogs and toads and Caecilians on Monday. I will see you guys in lab tomorrow. If you could be there at the beginning of lab, I want to talk to you a little bit about your dichotomous key. so that you're making progress on that, and I'm a little clearer in kind of how that's structured and everything. But then after that, you'll just be free to look at specimens as you want to. All right. Yeah, Rose.
Wait, so will you log on to Zoom tomorrow so that I could hear that also?
Oh yeah,
After
sure, I
giving
can
you
do
the
that.
lab?
Yeah, that's a good idea. I forget that because I see you and I'm like, oh, you're just here, but you're not here. Yeah, yeah. Let me do that. Let me make myself a note. All right. Yeah, I'll send out an invite, and it'll go to
Okay.
everybody, but Rose will probably be the only one who logs in that way. All right.
Okay.
Then I'll see you guys tomorrow.